Good morning. Good morning. We are going to move into our next presenter. Before I do that, I'd like to introduce myself. Many of you in the room probably know me, but for those of you who don't, my name is Nicole Williams, and I am the Assistant Executive Director for the Missouri Immunization Coalition. I would just like to reiterate what Nicole Cope said, and a big thank you to all of the planning committee members who helped make this event a huge success. And of course, this event would not exist without all of you traveling in to learn and network together. So we are, of course, appreciative of each and every one of you for being here. So without further ado, I am very honored to introduce our keynote speaker, Dr. Daniel Graciaa, who hails from my alma mater at Emory University. Dr. Graciaa is a clinician-scientist with a clinical and research training in infectious diseases. He serves as assistant professor of medicine at Emory University, where his focus is on clinical and translational research in infectious diseases, particularly translational immunology and vaccine clinical trials. During his medical education and clinical training, he received additional training in epidemiology and clinical and translational research, earning both a Master of Public Health degree and a Master of Science in Clinical Research degree. At the Hope Clinic of the Emory Vaccine Center, Dr. Graciaa serves as the site's primary investigator or sub-investigator on multiple vaccine clinical trials, including through NIH-funded networks. Please join me in giving a warm welcome to our keynote speaker, Dr. Daniel Graciaa.
Good morning. It's great to be here. I'd like to thank the Missouri Immunization Coalition for inviting me to speak with you today. I am Daniel Graciaa. I'm a faculty member at Emory, as mentioned, and I'm excited to talk to you about vaccines of the future. So novel vaccines and vaccine technologies. It's a pretty broad topic, so I hope to focus on a few illustrative examples of vaccines that show how new vaccines are being developed and what we may look forward to. So just a note of disclosure before I begin. So as mentioned, a lot of our research is funded by NIH. Obviously, anything I'm discussing is my own views, is not those of the NIH. Also, I am coming from Georgia, but I'm not a Georgia football fan, so please don't hold that against me. Whenever Missouri plays Georgia, I'm definitely cheering for Mizzou. So it's good to be back in Columbia, and I'm excited to talk with you today. So I work at the Hope Clinic, which is the clinical trials arm of the Emory Vaccine Center. Our mission is to address global public health threats by translating basic research discoveries into clinical advances. So we do that in several ways. As mentioned, we do clinical trials, vaccines and treatment, and prevention. We do translational human immunology studies. We do a lot of training, both of residents, fellows, postdoctoral researchers, and then we are really committed to community education, community engagement, and participation. The work that we do at Hope Clinic would not be possible without the community. So some of the funding sources that we have are listed here. I won't go through all of them, but just to give you a sense of some of the networks that we work with, the VTEU, which is the Vaccine and Treatment Evaluation Unit, the COVID Prevention Network, the HIV Prevention Trials Network, and the HIV Vaccine Trials Network, along with some other funding sources. So we have really a diverse portfolio of vaccine clinical trials, but also treatment and prevention trials that we do. So the VTEU is kind of one of our main cornerstones. That's one of 10 national sites serving as the clinical trial arm of the government for vaccine testing. And we have two main locations at the Hope Clinic and also the Emory Children's Center doing pediatric research, but also adult trials there as well. We have a really big team, and so the work that I'm discussing today is not possible without that team, so I'm very grateful to them. Nearly 20 faculty, 90 staff. We do a range of studies, vaccines, therapeutics, pharmacokinetics, human challenge studies, and then having preparedness for pandemics, so pandemic influenza studies, COVID, Ebola, and then other emerging and important infections such as Zika, chikungunya, multi-drug resistant organisms, preparedness for bioterrorism. We also have a clinical trials unit, which is funded by the Division of AIDS from NIH. This does clinical trials of vaccines and prevention for HIV. One of multiple sites across the country doing HIV vaccine efficacy studies, evaluating monoclonals for HIV prevention. And an important move from the HVTN currently is what's called experimental medicine trials, doing early phase 1 trials of HIV vaccine candidates. And I'll talk a little bit more about that later. We also do prevention trials, as I mentioned, biomedical and behavioral intervention prevention for HIV infection, including, for example, HBTN-003, which showed the superiority of using injectable prevention compared to oral PrEP. We also do translational human immunology studies. So we're one of multiple sites for the HIPSI, which is the Human Immunology Project Consortium funded by NIH. We do challenge models of human infection, influenza in this example. We have also done Shigella and other enteric diseases. And a big growth area for us has been doing tissue immunology, looking at translational immunology and sampling different areas where the immune system is working. So in the immune system, in the lymph node and the bone marrow, to get a better understanding of how vaccines are working, how the immune response is generated, and how we can design better vaccines that way. And a critical part is mentorship and training. I've been the beneficiary of this through my training, but we have a T32 vaccine training program that's graduated 35 fellows so far, most of whom have stayed in research or academia. We also have a residency training research program called STAR, and a new program for undergraduates who are underrepresented in medicine called INSPIRE, and really training across the spectrum of trainees and including ongoing education of our faculty. So that's a little bit about who we are at Hope Clinic. I want to give you an outline of what I want to talk about today. So we can't talk about new vaccines without talking about COVID vaccines, but I want to focus on both what we've done with COVID, as a country, but also how do we guide future vaccine design for COVID vaccines. One example of novel vaccine technology, so using a microneedle patch, and then a few examples of structural design for vaccines, and then a little bit about what we do with translational immunology. So we all know about the success of COVID-19 vaccines, the speed that these were developed, less than a year from the sequence being available to having an effective vaccine. There was obviously a lot of groundwork that was done before we had a vaccine. It was really preparedness that allowed us to do this so quickly. And it really allowed this enormous, successful immunization campaign, 13 billion doses globally. And it's estimated the US averted over 3 million deaths and maybe close to 20 million hospitalizations. And you all know that the primary series coverage for people, especially those in the highest risk age group over 65, was great. It was nearly 95% or higher, decreasing as the age bands decreased, as you can see on the right. I think the age labels disappeared, but the highest there is 65 plus and then decreasing ages. So the vaccines that were available until last year were the mRNA variant vaccine platform and then a protein subunit using the ancestral or original vaccine strain. And I think we also all know that despite the availability of these vaccines and recommendations for everyone to get at least one dose of them, there was pretty low uptake of the booster, over just 20% among all adults and less than 50% among those 65 plus. And there are a lot of reasons for this, but I think the important thing that we have to keep in mind in talking to patients is that most of the population, because of that, has some immunity to COVID. So this is a serial survey of blood donors done by CDC. This was published last year in MMWR. And the blue bars, the darker blue bars at the bottom of these graphs show previous vaccination combined with infection. So that confers what's known as hybrid immunity. And this proportion understandably grew over time from in 2021 through 2022 as people got vaccinated and also infected. And this is important because hybrid immunity protects against severe outcomes. So this is a modeling study that was published in Lancet ID, and the figure on the left shows estimated protection from different combinations of primary series or booster vaccines plus infection. So you can see the blue bars at the top. Those are hybrid immunity, so combination of infection and either primary series or booster vaccine. And those confer the best protection and estimated and shown now to be durable out to at least 6 months and probably longer. So better protection than just primary vaccination alone. And on the right, for any infection, you can see that's still true, although probably less effective protection and waning sooner. So these concepts of hybrid immunity and waning are really important for how we think about developing future vaccines for COVID. So one thing that NIH did to look at this was called the COVAIL trial. This was funded by DMID. It was a phase 2 randomized trial that was done in sequential stages, looking at different vaccine platforms among healthy adults who had received a primary series and a single booster dose with or without prior infection, and was done at multiple sites across the country. So there are several publications that have come out of this, but I'm going to focus on the top one here, which was looking at comparing the bivalent and monovalent vaccines and talking about antigen mapping. So this is an antigen map. So what's looking at how different antigens that are involved in infection and vaccination are related to each other. So if you kind of imagine you're looking top down and you see the different antigens mapped out here, different strains of the virus, the ancestral strain is over here on the left in the blue, circled in red. And then the different variants as they emerge as they are getting more distinct from the ancestral strain, are getting further apart. So you can see some of the Omicron variants like BA1 and BA4-5 are in the red, much further away from the other strains that are more similar to the ancestral. So the COVAIL study looked at different combinations of antigens and how they affect the antibody response. So that same antigen map that I showed on the previous slide is now here as kind of the baseline or the floor of this figure. And on the left, you can see those that had no previous infection had a boost in their antibody response. So these kind of platforms or the planes show the neutralization titer against these different variants. And they're plotted against the map in a third dimension here. So if the surface is flatter, it means there's less variability in the titer. So you can see there's a decent amount of variability among those who did not have previous infection. But from day one, which is the lower portion, and then day 91, which is higher, there was a decent boost in neutralizing antibodies. And then on the right, with previous infection, you can see that, okay, wasn't me, I don't think. You can see that they had a higher antibody titer at baseline, but they also had a boost with additional vaccine. And you can see that with the different combinations, those with the Omicron-containing vaccine had a higher boost compared to prototype. That's in the different colored text on the right there. So this is, I think, a good illustration or proof of concept of what was done with developing these new vaccines. So this is a little bit older data now, but I think still useful to know that we've seen from data published in MMWR that variant vaccines work. The vaccine effectiveness probably around 50%. Looking at different outcomes, symptomatic infection maybe a little lower than severe disease, but they have waning over time. And so this lower portion of the table here shows outcomes for hospitalization, which I think a key severe outcome. And you can see that at this point, and these are data from 2022-23, that the monovalent vaccine was not very effective, only estimated about 25%, but boosting restored that vaccine efficacy greater than 50%. But as time went on, out of 120 days or later, it had really waned substantially. So we all know as well that new updated vaccines were developed with the monovalent XBB or Omicron variant. And this is data from just showing that giving that updated vaccine boosts the immune response. So across variants, you have an increase in neutralizing antibodies from before and after. And we know now that these updated vaccines also boost protection against infection. So looking in different populations and different outcomes, at the top here is immunocompetent patients with an outcome of emergency department or urgent care, Again, vaccine effectiveness probably around 50% and waning over time. And then immunocompromised patients down at the bottom, looking at outcome hospitalization, still having some vaccine effectiveness, but maybe not as much as we would like, and probably some decrease over time, although a little early to say. So others are experts on immunization messaging. I am not, but I think that this figure from CDC was helpful for me in talking to patients about updated COVID vaccine and taking a illness that is pretty common and people may not perceive to be too much of a risk to them, but it can be, and we know that, but taking that from mild to mild, so preventing it from being a severe disease to hopefully something that's less serious. So we know that these vaccines protect against severe disease, hospitalization, and death, but we know also that previous vaccine protection has waned over time. And there's still, I think, an important goal of decreasing overall infection, transmission, hopefully, and emerging evidence for decreased post-COVID conditions from having COVID vaccine in terms of long COVID, which I think is an important concern. Despite all this information, we're still trying to figure out how do we best define what is protection from COVID from vaccination, and that's looking at a correlate of protection. So we've historically always used antibodies as a correlate of protection, and that was the thought too for COVID, but I think now we have good data that that's the case. There's probably more, we know there's more to it. It's not just about antibodies, T cells work in our immune system as well. We have other ways that our immune system protects us with vaccination. But I think this is a nice example of looking at antibody protection from COVID. So on the y-axis is the probability of COVID through days after vaccination. You can see the placebo is this gray line that just goes straight up. And then different tertiles or different components of vaccine antibody as the vaccine-induced antibody increases, then your probability of having COVID over time decreases. That's what the green line's going down there. And then this is vaccine effectiveness based on an ID50, which is a neutralization titer for antibodies. You can see with participants who had undetectable antibody, they really were not protected. But once you get to increasing ID50 value of just a 10 or 100, you get up to 78 or 91% vaccine effectiveness. And then with higher antibodies, better protection. So this, again, does not account for everything the immune system's doing to protect against COVID, but we at least now have good evidence for how we can compare future vaccines to existing vaccines for how well they protect us. So we know that those vaccines are good at protecting against severe outcomes, but what can we do better? So can we do better protection against infection, better protection against transmission? You've probably heard about Project NextGen, which is a very large government-funded program for COVID preparedness. The goals are to improve the durability of immunity, protect against emerging variants, but also other pandemic potential coronaviruses, as we've seen very clearly that this family of coronaviruses has huge pandemic potential. And then looking at other ways, either other delivery mechanisms or other vaccine design to decrease infection and decrease transmission. So I know everyone's tired of COVID by 2024, so we'll talk about some other stuff for the rest of the talk. Some exciting news coverage that you may have seen is new vaccine platforms such as the microneedle patch. And this has actually been shown in an early clinical trial in Gambia to look pretty promising and has been funded by Gates Foundation for production. So I'm not going to talk about the measles and rubella patch, but broadly about microneedle patch in the context of influenza vaccine, which is a study we did at Hope Clinic. So I don't have to tell this audience that influenza vaccine uptake is not great. And there are a lot of reasons for that. But I think the ones that microneedle could have an impact on are both patient factors in terms of patient concerns about needles, needle aversion, adverse effects, and time related to getting a vaccination. And then there are system issues in terms of storage, cold chain, cost of administration. And so the microneedle patch is just that. It is microneedles or a dissolvable component that is on a patch. So it's basically like a bandage that you apply to the skin, leave it on for a period of time, about 15 or 20 minutes. And the vaccine antigen is delivered across the skin. And the benefit there is there's actually immune advantages because there's a lot of antigen presenting cells in the skin. And animal models actually showed that you have very good immune response as a result, and maybe even potentially better than intramuscular vaccine. So the example that I'm going to talk about is for a flu vaccine patch, which was given to 100 participants, or this trial involved 100 participants. So 25 people got standard intramuscular injection, 25% got microneedle with the flu vaccine, 25 got self-applied of the same, and then 25 got placebo. So that was an important component to see if that might improve acceptability for a participant to be able to give their own vaccination. I think the main question people ask when they hear about this is, do patients like it or participants tolerate it? And the answer is yes. The reactogenicity is similar to intramuscular vaccine, mostly mild. Obviously, patches are applied to the skin and they're having an immune response in the skin, so you're going to have some more reactogenicity on the skin in terms of some redness or itching. But most of that is temporary. It lasts maybe a few hours or a day. And then the systemic reactogenicity, the things we usually think about in terms of fever or fatigue, that was similar to the intramuscular. We showed that this patch is immunogenic, so this is showing immune antibody responses. On the left, overall, you can see that the seroconversion, which is on the bottom portion of those colored bars, and then the neutralizing antibody titers on the top portion are similar, and the blue, orange, and green are the different formulations of the vaccine, with the far left blue bar being the intramuscular. So the orange and green are the patch. And you can see they're pretty similar to intramuscular, and the purple is the placebo, so that's why it's lower. On the right is showing change in their titer against hemagglutinin. So the darker bars are the intramuscular vaccine, and the lighter bars are the patch. So you can see that they have pretty similar fold change in their neutralizing titers compared to intramuscular for the patch. So there was a survey component to this study, so the important takeaway there is that most participants said it was an overall positive experience. Most of them said it was better than getting an injection, and they would prefer it to getting a future injection or an intranasal spray. There are some limitations to that. It was only 100 people overall. You might get people who are less interested in getting vaccines to begin with, getting injections. But I think beyond that acceptability, we know that there's potential immune benefits, or at least equal immune response to intramuscular delivery, and potential cost savings in terms of manufacture, cold chain storage. It's still early for this technology, I would say. There are clinical trials now looking at, or that have shown some evidence for benefit, and then looking at different vaccine platforms or different infections. MMR I mentioned, but also flu and COVID. As this is scaled up, then the dosage is going to need to be standardized. And then manufacturing and storage, since it's new technology, will have to be kind of ironed out. But some exciting developments there in terms of 3D printing, developing thermostable microneedle patches that may make it much easier to distribute and apply these vaccines. All right, so wanted to talk a little bit about RSV and HIV vaccine, really as examples of structural-based vaccine design. So using the protein structure for an immunogen to develop a new vaccine. RSV has been described for a long time, and since it was identified as the cause of bronchiolitis, work started to develop a vaccine. There was actually an inactivated vaccine candidate that was developed in the 60s, and unfortunately did not do well. It actually worsened outcomes among infants who got the vaccine. And so that really slowed down vaccine development for RSV for a long time. And there weren't really prevention measures available until a monoclonal came out in the late 90s. We know that this is a very common infection. Up to 3% of infants are hospitalized in their first year of life, and it has significant burden for adults as well. substantial hospitalization and death. And as everyone in this room knows, we have new vaccines finally as of last year, a couple of vaccine products and a new monoclonal antibody. So these vaccine products are given to adults, and then the monoclonal is given to infants and young children. But the story of the development of this vaccine is, I think, pretty interesting, again, because of the time it took, and also the kind of persistence that it required from folks at the Vaccine Research Center at NIH. So the protein was first isolated, in 1989, and despite that, it still took a long time to get to a vaccine. So they had found that this certain conformation of the protein was the target of these neutralizing antibodies. And they still had to really define the structure and then be able to stabilize the structure in a conformation that was useful for a vaccine. And that was done in 2013. It was one of Science's top 10 breakthroughs of the year. And that group continued working on this and used the Middle East Respiratory Syndrome coronavirus to define the spike structure for that. And that provided a platform for when SARS-CoV-2 emerged, they could quickly develop a vaccine. Having that structure allowed the development of the new RSV products. So HIV vaccine development is, you do have whole conferences on this, so I'm not going to pretend to cover everything that is relevant in this in a slide. But I think it's another really important immune and vaccine challenge that's been worked on for 30 years. While there's been multiple efficacy trials, only one showed modest benefit and it wasn't sustained. So the current strategy is to try and induce broadly neutralizing antibodies in people who receive vaccines, so in a couple of different ways. But again, using protein structure design to target the germline, meaning like early on in the immune response, how do you start antibody development that then you can guide with future vaccines in a kind of prime and multiple boost regimen to develop broadly neutralizing antibodies. The reason this is being focused on is because, as I mentioned earlier, some of the passive immunization studies that were done in adults showed proof of concept that if you give neutralizing antibodies to people that are preventive of HIV infection, but only against vaccine strains that are susceptible to that specific antibody. So we need broader antibodies that can neutralize multiple strains of the virus. There are a lot of factors, there are a lot of challenges in HIV vaccine development. So that includes the viral diversity, as I mentioned. And I think a really important one practically for HIV vaccine efficacy trials is now we have very effective prevention measures for HIV, so in terms of PrEP, oral and injectable. And so that makes it difficult to evaluate vaccine efficacy in a trial. But still an important avenue that is being worked on at Emory and other places through the HVTN. And then some new things that you may see coming down in the news are new strategies in terms of vaccinating people who already have HIV infection to see if they can identify neutralizing antibodies that way. And actually having people on effective treatment, holding that treatment and evaluating their immune response while they're off therapy to see, again, to identify effective vaccine targets. And lastly, I want to talk a little bit about translational immunology. So it's a broad concept, but the idea is to really use systems biology approach. So that means trying to understand the full immune response in the host to a vaccine or an infection. And so that's looking at genetics, the microbiome, environmental factors, and the host immune response through a variety of techniques to identify how our body generates an immune response to vaccine, and with the goal of identifying something that could predict that vaccine response and then inform better vaccine development as a result. So I mentioned a little bit about tissue sampling, so I think this is really the future of vaccine research. We have for a long time relied on using peripheral blood to evaluate the immune response because it's accessible and much easier to get than sampling an immune tissue. But we know the immune system works not just in the peripheral blood, so it generates a response in the bone marrow and in the lymph nodes and in other places too, but these are kind of areas we can target. And the magnitude, the durability of the immune response depends on the process of B-cell development throughout these compartments. And the vaccine response as well also depends on a variety of factors such as the dose, the route of administration, the type of vaccine, and then other adjuvants, for example. But if we can better understand how the immune response works in these compartments, we can hopefully design vaccines that produce more durable immunity. So one example of this, using bone marrow aspirates from investigators at the Emory Vaccine Center, was showing bone marrow plasma cells generated by flu vaccine, demonstrating them in the bone marrow, but showing that they decline a year after vaccination. So kind of initial proof of concept of doing this immune response in the bone marrow with the goal of then designing a better flu vaccine down the line. So I mentioned these different sites, and this is something that we do at Hope Clinic. Obviously still our old standby with peripheral blood, but using lymph node sampling, so either through fine needle aspirate or biopsy, or doing bone marrow aspirates. So we've done over 40 of these lymph node samples just since January, and over 50 of the bone marrow aspirates in the last year. And this is one of our study exam rooms with the ultrasound machine, so we were able to do these procedures in our research clinic. They're well tolerated. People have been, we've had no complications, honestly, in the time we've been doing these. And we're using this to look at multiple studies for different vaccines or prior infections. So that's flu, as I mentioned, but also yellow fever, HPV, which may inform the discussion about how many doses of HPV vaccine are really needed in terms of durability response. A lot of places have rolled out just doing 1 dose of HPV, whereas the standard has been 3. Ebola vaccine, immune response, and then preparedness for other vaccines or other infections as they come. So to summarize, we talked a little about COVID vaccines as an example of vaccine development, but also for how do we make those better. Novel vaccine technology and design is still needed, both for existing infections and also preparedness for future ones. And then we can use translational immunology studies to inform future vaccines. I put the picture of the eclipse up there because the recent eclipse got me thinking about the last one that was close to Georgia in 2017, how different the world was even just 7 years ago in terms of infections, not even knowing what COVID was, not having COVID vaccines, obviously, and how quickly those things were able to come to be. And the next eclipse, I think, is 20 or 30 years from now. So just thinking about the future vaccines and how different things may be at that time, but I think we have a lot of tools that we can use to move things forward, and I think it's really exciting times for vaccinology. So I'd like to thank you for your attention. I'd like to thank Dr. Rufael, who's the Executive Director of the Hope Clinic, and the whole Hope Clinic and Emory Vaccine Center team. As I mentioned, it's really a team effort, what we do, and the community that supports us as well. And I'd like to thank you for all the work you do for immunization coverage in Missouri, and I know the challenges that we're experiencing as a country for vaccine coverage are similar in Georgia too, so thanks for the work you do, and thanks for your attention.
Thank you, Dr. Graciaa. It can be easy to get bogged down in our day-to-day work in immunizations, but I know I am at least inspired hearing about what's on the horizon for the future of vaccine technologies and new vaccines that are soon going to be available for all of us to help keep Missourians safer. So thanks for being here and sharing that really inspiring sort of where we're going towards the future of vaccine technologies and new vaccines. Let's open it up to some questions now from the audience. Anyone have any questions? We will pass around the mic. Yes.
Good morning. Can you tell me if the storage and handling for the patch stuff is the same as the storage and handling for the syringe vaccines?
So I think that's one of the potential advantages for the patch. The designs that are being worked on are thermostable, so they wouldn't require a cold chain. And if they are kind of implemented as hoped, they could be stored just kind of, as long as it's in a dry environment, but they could be packaged in a way to protect them, as you imagine other medical products that need a dry environment. So I think there's a lot of potential storage and transport advantages to patch coming down the line.
Other questions? I see a hand up from Dr. Irvin over here.
Hi, I just wanted to know, are there any differences in contraindications or precautions with the new administration technologies? Specifically Guillain-Barré or allergic reactions?
Yeah, I think that's an important question. Thanks for that. I think we've seen already with RSV, there was some safety signal in the early studies about maybe neurologic events such as Guillain-Barré, and there's a little bit on that on post-implementation surveillance. So I think that's why it's important that we keep those systems in place to monitor for that. As far as, for example, I didn't talk too much about intranasal vaccines, but there is an intranasal flu vaccine that has some different contraindications as far as compared to intramuscular, people with respiratory symptoms or respiratory conditions or immunocompromise not being able to receive those. As far as the patch, the studies so far I think have tried to exclude people who have significant dermatologic conditions, so severe eczema or other things that may have skin breakdown in an area where the vaccine would be administered. So I think that'll be an important part to keep in mind as well. But I think that's the benefit of having multiple avenues or multiple administration routes is being able to have something that's accessible to people who may have contraindications to a certain platform.
Dr. Graciaa, thank you for a really fascinating talk. Regarding RSV vaccines for infants, right now, the things that we have, which are amazing, are passive. Either transplacentally because the mother got vaccination or giving antibodies to the baby. Is there any work being done on an active, vaccination for infants that would provide a longer-term immunity?
Yes, I believe there are studies going on that. I think, as you pointed out, the passive immunization, we know it works, but it has administration challenges, and I think also important for all vaccines to keep in mind equity and how people can access them. And so making sure that we're protecting as many infants as possible. I know that the previous experience with active immunization in infants appropriately had raised a lot of caution in terms of development. So I think it's a slower process than we might expect otherwise. But I hope that with these newer platforms that have been developed that that might be something coming in the future.
Other questions from the audience? We're looking around. I've got one. So you've talked through and you walked us through lots of examples of what's on the horizon. Looking forward towards the future of immunizations, what's one thing that you're most excited about in the research that you're doing and that you've shared with all of us today?
So I'm biased because that's what we work on the most, but I think the translational immunology studies, not just from a academic question, but I think the collaborators that we have who are at Emory, but also other places, so Bali Palendran, who's at Stanford, they've identified a vaccine signature, they call it. So basically something they can look at baseline in a participant, and based on their, and shortly after vaccination to, as a marker of how their immunity is gonna respond to that vaccine. So I think that is very exciting in terms of how we can develop vaccines broadly, but also potentially make sure that we are ensuring people who receive vaccine are actually getting the protection we expect for them. So right now, most of the time we rely on antibody response for certain infections. But I think making sure that people who receive a vaccine are actually protected as we expect them to be is very important for both, I think, for a population level, but also for communicating with patients and ensuring they're getting the protection they expect.
Absolutely. Another way to double check that they're protected in the way that we anticipate.
Yeah, absolutely. And I think it also informs how to design other vaccines, because if you can rely on this platelet signature or other signatures they may identify, then as you're developing new vaccines, can you then maybe be more confident that that vaccine candidate is a promising one to evaluate.
Excellent. Thank you. Any last questions from our audience?
I have a question. Oh, go ahead.
Okay. Well, how about we do here and then we'll pass over.
I was wondering, for the patch, do they have certain places on the body where they're trying to administer it, or can it be pretty much anywhere?
So the flu vaccine patch that I described was placed like on the dorsum of the hand, on the back of the hand, or on the wrist. I think generally they try and keep it on the extremity, so it's kind of easy to evaluate for reactogenicity response. But I think it would have potential implications to be administered almost any part of the skin. I think for standardization purposes, they'd probably try and keep it as similar area because of dose delivery and thickness of the skin in different areas. So that's why they kept it consistent there. But yeah, that's a great question. I think there would be some flexibility there as well.
So my question, you may or may not have information to share with us because you didn't mention it, but I recently read an article actually just this week about the potential for a universal vaccine and the research and development around that. Do you have anything you could potentially share with us about that?
Universal flu vaccine, or yeah, so that's something that's been, I think it's kind of the holy grail of flu vaccinology. It's been worked on for a long time. Specifically in trials that we have at Hope Clinic, we don't have that going on right now, but I know there's something worked on at the Vaccine Center. I think it's a particular challenge because of, or the goal of, the reason for doing that is because we know the flu strains change every year, and we have to go through this annual exercise of predicting strains and how to best protect against them. So I think it's exciting potential, but yeah, I don't have specifics to share on it right now.
I know it's very new.
Yes. Yeah, I think it would, if they're able to successfully do that, I think it would also help us with other viral diseases, in terms of COVID as an example, where variants emerge, and being able to provide long-lasting and effective protection against a variety of variants.
Any other final questions from our audience before we finish up? Seeing none, let's give our keynote presenter one more round of applause.
Thank you.